Good morning and welcome everybody to the webinar Discover a New Era in Microscopy. I am excited to introduce our speaker today, Dr. Falco Kruger. But first, let's introduce me. I'm the host. I will be your host today and I will take care of issues. And if you have questions, we will handle them at the end. Falco has been working at Leica Microsystem since 2018, and he has been focusing on advanced widefield microscopy. Today, he will be presenting an exciting new innovation in microscopy, Mica, the first imaging microhub. This new system promises to revolutionize the way we conduct microscopy and will be of great interest to anyone who works in the field. Falco's expertise and experience make him the perfect person to lead this webinar and provide insights into this groundbreaking technology. I also would like to mention that the Mica has received a Red Dot Design Award, which is an internationally renowned design competition that rewards outstanding design quality and innovation. In the opinion of the jury, Mica distinguished itself in terms of design quality and degree of innovation. Like an airport hub that brings together passengers and guides them to their destinations, the world's first imaging microhub, Mica, guides the users to their experimental destinations regardless of their microscopy experience levels. But before we begin, I would like to inform you that this session will be recorded and the recordings will be sent to you after the session. You can also access all our past and upcoming webinar recordings on our website under the events section. Additionally, a certificate of attendance will be sent to you after the session, and we will answer your questions at the end. You can submit them at any time using the key function at the right-hand side normally. But lastly, we would greatly appreciate if you could fill out a survey at the end of the session. Your feedback is essential for us to improve and provide you with the best possible experience in future webinars. So let's welcome Falco to the virtual stage and hear all about this innovative new system.
All right, then I will hit my magic start button. And good morning, everyone. Thank you very much for this introduction. My name is Falco Kruger. As it was already introduced, I'm advanced workflow specialist. I also lead a small team. And currently I am broadcasting from our Leica labs at the EMBL Imaging Center in Heidelberg. So if everybody or anybody is close, hello and welcome. Behind me, you can already see the star of the show. And maybe as a small interesting note, something that I need to be very open with you about is that today is actually my 125th demo with that exact system. So I started last year in March, actually, when we introduced Mica as the world's first microhub. And today I'm still excited to present to you why we created Mica. What is the main idea behind Mica? I mean, a little bit was already mentioned to you. I'm also surprised this morning, actually, the Red Dot Design Award was given to Leica and for the Mica system. So we will also have a closer look at the nice design features. But most importantly, what you need to understand today is that there are three main things we want to achieve with Mica. And the first one is access for all. And access for all means that everyone in your lab, in your team, is able to work with Mica. Okay, why is that possible? Well, independent of the imaging experience, and you might know, if you are an imaging expert, you know how long it took you until you felt safe in order to optimize imaging parameters, how to set exposure times, how to set laser intensities and detection windows and all of these kind of things until you became an expert. And the cool thing with Mica is that the intelligence or the experience that you need in order to optimize imaging parameters are in the system and not required by the user anymore. What the user needs to do is basically mount the sample, type in what kind of fluorophores are inside the sample, and then start imaging. Everything else Mica will take care of. And not only this, the second main important message is that Mica provides or has no constraints when it comes to the imaging modalities it offers. So the system behind me actually can do wide field imaging, thunder imaging, confocal imaging, enhanced resolution confocal imaging, and also your classic H&E stains and so colorimetric imaging as well. And Mica is an incubator, so you can do all of these things also under environmentally controlled conditions, right? So all of this is packed into a single box type system, yeah? And that's also why we call it a micro hub, not because it looks like an airport, but because all of the different standard imaging workflows that happen every day in every lab can be combined into a single system, okay? And this paired with an ease of use that has never been present before, yeah, you have a much higher throughput and yeah, have much more robust results. And the third main thing is actually that we provide also a certain level of automation and intelligence that guides you and makes it more convenient to work with Mica. All right, so Red Dot Design Award. Maybe I should start to talk in the beginning a little bit about the outside of Mica. Basically, what you see here is a white shiny box. The front actually you can use to draw with marker pens on it, and then you can wipe it again. You can leave messages or just write "do not touch," experiment running, stuff like this is possible. Mica sits on a passive anti-vibration table that's part of the package as well. And if you can see it here down below, there is a small box, and in this box we have all the light sources. As I mentioned before, we are doing wide field microscopy and confocal microscopy in one system. So we have 4 individual LEDs to excite your fluorophores in wide field mode, and we have 4 independent diode lasers that excite in confocal mode. So you really have 2 distinct pathways, really a mixed microscopy system here. If you have a closer look in the back, the small backpack over here is the system for the environmental control, okay? You don't need anything more. It's basically just also in the field, if you want to upgrade it later on, it's just attached to the back of Mica, connected to your wall mount of CO2 or your bottle of CO2, depends on what you are using. And you can also do live cell imaging. That's basically from the outside. Everything that yeah, you see is basically part of the Mica system. You open up Mica by clicking on the open button. And why do I have a torchlight with me? Well, basically to have or create also a nice look for you and a nice contrast in the inner part. I will also quickly change to a different magnification so that you can also see something. So what you see here is that Mica has a very clean design. As I said, we intend also to provide multicolor fluorescence imaging yeah, capabilities to any and also inexperienced users. So there's nothing that you need to fiddle around with, okay? So Köhler illumination, the system comes completely calibrated, okay? That's why we have here a box around it. So there's no need for you to check whether you have the perfect illumination because Mica comes from the factory like this. And there's also no access that you can basically change anything. Here, those are basically the vents for the live cell incubation, and something super interesting is this small port over here, which you can open up and then guide tubings for microfluidics experiments inside of the inner volume of Mica, because this entire volume that you see here is the live cell incubation chamber. Mica has very thick and highly insulated walls, so the environmental conditions are extremely stable over long periods of time. Really, several days of live cell imaging experiments are possible. And of course, if you want to change samples, you don't open up the big front of Mica. What you can do is actually, oops, open the front flap. Okay, so here you also get a very short access to Mica. You can pipe it during an experiment, pipe it things into your experiment, some compounds that you want to test, whatever you want to do. Close the lid and you have minimum loss of environment or of your environmental conditions. You can also use this here as a shelf, so you can put here a medium bottle or pipette tips, whatever. It's really to have close and very short ways of working. Yes, and that's basically it from the outside of Mica. What is really more important now is, okay, how does Mica perform? And unfortunately, I have to say that this morning during startup at the 125th demo that I want to do with that system, the stage actually quit working. It still moves around, but not reliably, and that's why it needs to be exchanged. The technician is already ordered, but I will do my best to show you all the individual features that Mica offers, also for your work. But just keep in mind that sometimes I will try to avoid a certain movement just to, not create weird images. But we will do imaging. All right. Let's have a look into the surface. What you see here is actually already the second step. This is your working interface. I made it a little bit smaller that you can just appreciate in the beginning. Okay, here, this is the sample that I have mounted. All right, and that's the user interface that you have with Mica. Usually, you would start with a sample definition. So sample definition is the first step that is required. You as a user, no matter how experienced you are able to tell those main features about your sample. So you start here on the top left corner by simply telling the system what kind of experiment are you doing, and then you specify what kind of carrier type do you have. So I have a simple glass slide, okay, where on top there are two round cover slips and where we have fixed cell culture on it. So the cells were grown onto the cover slips and then stained, or fixed and then stained, and then they were mounted on those glass slides. So they are adherent to the cover glass. And this is actually what I did here. So you type in characteristics of your sample. So if you have a cell culture grown on the cover glass, those samples are adherent. Detached would be something like you have a thick tissue section, right? I don't know, 20, 30, 40 microns. And you know that if you mount them on the glass slide, sometimes they can be a little bit wavy. In the end, what it means is you not always have the same focus plane, right? And what you are specifying here with these characteristics is basically what kind of focusing method Mica is applying. There are two main focusing methods that you can do, and this is software autofocus and hardware autofocus. Software autofocus is your classic, I record a volume and look for the sharpest image in that volume data set. And the hardware autofocus is working with a small light beam that is reflected from the cover glass and then detected. So to keep the objective lens always in the same position with regard to the cover glass. So even if your sample is a little bit tilted, the objective will follow these slopes. Of course, you could also image 96 well plates, 384 well plates, imaging dishes, and also something that is very nice in combination with the imaging speed of Mica. We now also offer 4 slide holders that if you have several slides to image, you simply put 4 of them already into a single insert and then prepare your experiments. The most important thing and the most tricky thing that you need to do with Mica is to type in the dice. Yeah. So the only knowledge that you need to provide as the scientist working with Mica fits on a sticky note, right? What I've written down here are basically the dice that have been used in my fixed cell culture, and these are the structures that have been labeled. And that's the only thing that you need to put in order to tell Mica what kind of multicolor experiment you want to do. And this is what I have done over here. So in staining, I have simply selected the 4 dice, Höchst, Alexa 488, SPY 555, and Atto 647. These are labeling different structures. If you want to give a name to a channel like Höchst would label DNA, you can simply type in here DNA and you do your imaging. That's it. Okay. So how Mica works is, instead of using fluorescent filters, it's using the excitation and emission profile to know what kind of fluorophore you have. That's really amazing because if you're now using Höchst or using DAPI, sometimes you might want to use a different fluorescent filter, yeah, in your classic microscope systems. For example, if you go for red dyes, you can have an MRFP, an M Sherry, a Texas Red, and they all are called red dyes, but basically their emission spectrum is completely different. So what you would need is different fluo cubes, so fluorescent filter cubes. With Mica, you don't need this. With Mica, you simply specify what kind of fluorophore you are using, and that's it. Okay, so you create an overview, click here on overview, and this is what you get, an overview image. And then you end up in this user interface. In the center, what you can see here is the stage area. Okay, like in Google Maps, for example, you can zoom in and out simply using your mouse wheel, but you also have your small mini map. Okay, so that even if you are highly zoomed in, you always know where you are in your sample. And on the left-hand side, you see how you are guided through creating an experiment. So you start with magnification. Here I'm now starting with a 20 times objective. Then a very interesting mode or an interesting button, widefield and confocal. Okay, this is the only thing, and you will see this later on. The only thing that you need to do is click on confocal and you switch from widefield imaging to confocal imaging. Here we have transmitted light functions, and this is also where I now start my experiment. I simply click on live and fly into the position where I am. And what you can see here is this is IMC, integrated modulation contrast. So I'm not going into the technical details. What I want to present to you here is if I zoom in now also digitally, what you can see nicely are subcellular structures. So you really get a three-dimensional impression, even though this technique is not DIC, something that you might know already. The advantage of IMC over DIC is that you can also use it through plastics. I don't know if you are usually using phase contrast or DIC, but phase contrast you can use through plastic. If you have, for example, a 96 well plate and you have a plastic lid on top, you can still use phase contrast, but no DIC. With this integrated modulation contrast or Hoffman contrast, you can also do this kind of 3D, or you get this kind of 3D impression from samples that have, for example, a plastic lid on it. All right, but now just imagine you're sitting in front of your system. So we have mounted here a sample, a cell culture, so a pretty easy specimen. You have mounted a sample and it has 4 colors. So how do you set up an experiment? I told you one thing that you want to do is you want to give access to all. So no matter how experienced you are, you can set up and do multicolor experiments. What we also want to do is to radically simplify workflows, meaning instead of now, as probably your system does at the moment, you set up channel 1, channel 2, channel 3, and channel 4. You select the respective filter cubes, and then you go into each channel and optimize or try to optimize the exposure time, the LED or the excitation intensity, all of these things. With Mica, what you need to do is you click live. All right, so you have specified the fluorophores, yeah, as I have shown here on the sticky note, but that's it. Okay, that's all the knowledge Mica needs in order to set up for you this multicolor experiment, and not even simply setting up, but also optimizing. But what does it mean, optimizing? Well, if you now have a look here, you see, yeah, the quality is a little bit grainy. Yeah, it's not the highest quality. You see here all 4 channels are already active. But you also see here under intelligent imaging a slider between sample protection and image quality. And this image quality is not at the maximum. Okay, so if I now want to have a higher image quality, I go here to 4, for example, then Mica is automatically adjusting all 4 channels for me. And you see now the image is much more smoother, the contrast is higher. And this is what the so-called one-touch button does. So here you could also click one touch, and then Mica is adjusting the imaging parameters. Something that is really, what I think is really amazing is, and I don't know if you've realized it, if I now move the stage, you see a multicolor image already, right? So usually if you, for example, create a spiral scan where you want to see where are my interesting cells over here in my sample. So it could be a tissue section. Right now we simply have a cell culture. But in order to find a nice spot where you can image or where you have your point of interest, this would be quite tricky to do. With Mica, you can adjust basically the image quality and have a nice view in 4 colors. If I now split up all 4 channels, you see here, this is the host channel, the green channel labeling the mitochondria, actin, and tubulin. Okay, if I now go to live and I move the stage, you see that all 4 channels are moving at the very same time. Okay, and this is the cool thing about Mica. It's not only very easy to use. Okay, I will repeat this quite a lot of time today in this session. But what we do here is simultaneous image acquisition, not a very fast switch between 4 channels. It's 4 camera detectors working at the same time. Okay, that's what we do. We excite the fluorophores, all the fluorophores in your sample during one exposure. This exposure right now is 100 milliseconds long. And we separate the light that is coming from the sample onto 4 individual detectors. And then based on the knowledge of the fluorescent dyes that you are using in your sample, we unmix those channels in real time. So what you see here is a 4-color image recorded at once, okay? And this makes all the multicolor imaging experiments so much faster, because one shot, 4 channels, okay? And with the current setup that I have here, what you can do is you can image up to 25 frames per second, okay? 25 4-color frames per second. So basically 100 images per second. And if I now create here a tile scan, for example, I click start, I simply want to demonstrate this to you. You are recording a tile scan, an overview with 4 colors at once, all right? So you're getting an idea much, much faster. And simply by clicking on live. So everything, I didn't touch any LED intensity, I didn't select any filter cubes because there are none, all right? Good. Your question might be now, okay, what happens if I have a dye that is not listed in the database of Mica? Well, you simply can teach Mica a new fluorophore by simply adding the excitation and emission profile of the dye. Yeah, and this you can download from openly accessible web pages. So that's also a very, very cool thing that you can do. And all right, so what we have created is an overview. We created an overview, and now we have hundreds of cells where we can look for, okay, where is the one exact cell division happening or cell division state that I'm interested in. And I don't know about you, but sometimes too much choice is also inhibiting me. Yeah, why don't we go here? So you double click and the stage jumps over there. I remove here that overview, and now I'm switching to the 63 times water objective. Okay, I stay seated because the water cap in Mica is not only automated, so it pumps automatically water, but it also measures the water capacity. So only if more immersion is needed on the objective lens, Mica will do this automatically. Okay, so for you, it's really the case that you put in your sample and you sit in front of the monitor and start working. All right. So I go to live. Here now, I'm a little bit out of focus. Something that I want to present to you is also the refocus position button. So basically an autofocus button that gives a very nice idea of, okay, how Mica operates during focusing operations. And this is by first doing a coarse scan of a volume and then having this first hint where might be the highest resolution or the highest contrast. It's then doing a fine scan. And this is what is happening here right now. And we now have a closer look of that position. Cool. All right. That's a very nice view here of cells. We have a little bit of cell division going on. We have here happily grown cells. We see also some actin fibers. And of course, now we want to have a look at, I don't know, some easy experiments that everyone in the lab does. So if you follow here that red line, we have set magnification. We are still in widefield mode. Simply clicking on live is giving us access to fluorescent multicolor experiments. And now we want to add volume imaging. Okay, so you click here on the 3D button and you have seen this. I just uncheck this again. See that you have here a small set of icons that you can use. And I don't know, for those of you who have already a little bit of experience with microscopy, basically what you usually do when you set up a Z stack, that's what it's called, this volume imaging, you click and define the start button or the start point, and you define the end point really on top of the cells. All right, Mica is automatically setting up here the view, and I click on start. All right, so again, recording 4 images at a time, we only move once through the Z stack. Okay, this is already done in just a handful of seconds. I couldn't even finish my sentence. We have imaged 27 slices with each 4 colors. All right, then we can have here a look at the results. That's basically here what we have just now recorded. Okay, that's very interesting. Here we have also the overview, the merged overview that we have created. And something that is very important, in the beginning, I was just calling all different names of what kind of imaging modalities you can do with Mica. I said widefield imaging and thunder imaging. And thunder imaging is something hopefully you know already. If not, I'm now giving you a brief introduction. The big problem with widefield microscopy is that you see it right now here on the screen. The better the optics of the objective gets, all right, or the properties, the optical properties, the more fine and concise the focusing volume is. Okay, this means that you are more and more collecting light from the sample that is not in focus. So basically, it's hiding all the structures. And this is what is happening here right now. I can move through the Z-stack, but basically what you see is a lot of blur. Only in a very distinct position, I can see here a little bit more structure, but that's it. So what thunder does is it removes the out of focus blur from the individual sections. On the right-hand side, you can see now each and every channel individually, all right? And what I do here is I go to Thunder, I select the raw data set, that's what is always recorded, and I click here on the small icon, that's why you call it Thunder. So you have here a lightning striking at that moment, and of course you would expect to hear a thunder. So what I do is I click on Apply, and just in an instant, the entire images are being cleared of background blur, all right? But that's not the only thing that you can do here. I'm going back to the raw data, selecting the second method of thunder, just clicking Apply. What is happening now is that you might have heard about imaging improvement functionalities like deconvolution and I don't know, background subtraction and all of these kind of things. So what we are doing here is currently a deconvolution, a so-called widefield deconvolution, where the classic thunder method removes, so subtracts background fluorescent, so not focused light from your image. Deconvolution is doing something differently. It improves also the contrast in the image, but not by removing out of focus blur, but by recalculating the position of this out of focus blur. Because in the end, this is stray light coming from a certain point in your sample. And this is what deconvolution does. It tries to bring back the stray light into its point of origin. And this is what happens when you are doing deconvolution. In rough terms, for all the experts, I'm sorry if I'm now breaking it down too much, but I would really like to follow, or everyone to follow. So, and then there's this third method. The third method is the logic consequence of doing either removing background with thunder or doing a 3D deconvolution. It first removes the background and then does a 3D deconvolution. So a lot of processing, which, by the way, you could also just simply activate while doing your imaging. You can have a so-called live thunder view, or you can simply, when you do an experiment like doing a volume scan or a tire scan, you can simply have thunder running in the background, because thunder operates in real time. As the images come in from the system, you get your raw data set and your thunder data set at the very same time. So you basically don't lose anything, you only gain more image information. All right, so enough talking. Falco, just show us the images. So this is the first method. Left-hand side is the raw data set, right-hand side is the thunder data set. Let's say the first level thunder data set. Okay, you see that much more, you see now really, you can follow the focusing through the cell in this overview. If I now go to small volume computational clearing, you see sometimes deconvolution can be a little bit harsh, but what I want to stress here is, I will zoom in a little bit more. The green dye, yeah, as written down here on my sticky note, is labeling TOM20. And for those of you who don't know this, TOM20 is the transporter of the outer mitochondrial membrane, so a subcellular organelle, and only the outer membrane of this. And where you can get already some kind of idea in this raw widefield data set, you can see nicely in this thunder data set that really membranes are being labeled, and only the outer membranes. You are creating holes when you are doing cross sections through a mitochondrium, and we are only doing thunder imaging, and thunder imaging is not even yet confocal imaging, but it's extremely fast and provides already a very high detail of your sample. As a third method, we have here, as I called it, LVCC. This is large volume computational clearing, the third thunder method. And I think you can really appreciate, for example, if we remove the mitochondria here, you can see this cell division event. You can see here the tubulin fibers in this, is it a centriole that is pulling away the condensed chromosomes during the cell division. So you see already in the widefield mode, with using the Thunder method, individual fibers of tubulin pulling apart chromosomes. All right. But especially during this case, and I'm lucky with this field of view. So this is now, let's say, a cross view of the cell division. If we move a little bit down, we have here the side view. Okay, super interesting. Today I'm lucky, except for the stage. So what you see here now is the side view, basically, of this event. Okay, this is not scripted. It's just yeah, it's nice that it happens. What you see here is basically now a point where improving the image quality of the raw data set with the Thunder functionality is not giving us the last bit of resolution that we need for the sample. Okay, so what would you do when this happens to you when you're sitting in front of your widefield microscope? If you are in a university environment, you check the, I don't know, image facility system calendar or booking calendar to see, okay, when is the next time I can go to my to a confocal system and then have a higher resolution. If you're working in a company environment, probably you also need to check when is the next confocal system free, and then you go there with your sample and hope to find this exact situation back. Yeah, this exact cell division pair here. Okay, of course, it's very unlikely that you do this because there are hundreds and thousands of cells, at least in my sample, that we have a look at here. So with Mica, as I said, we are bringing in different imaging modalities. Of course, it makes sense to do this because in cases like this, and this is not only a cell division, this could be something you are in your kidney section, you're in your organoid section, and you see a nice overview very quickly with the Thunder imaging, but now you need to segment nuclei, for example, in 3D. Yeah, and this last bit of resolution you achieve through confocal microscopy. And with Mica, what you do is you go to your imaging and say, okay, I'm interested in this cell division event over here. So what you do is you click on confocal. Let me just show you, or what we are going to do next is we are focusing on this cell over here. All right. And oops, the cell over here. Just focus down a little bit so that we can see here. All right. These cells. It looks oversaturated, but simply because we are here overlaying 4 individual channels. All right, and now I click confocal. So I activate confocal imaging, and I'm not yet going all in terms of resolution. So here I'm now just using the resolution of the cameras that we have been using before, and I'm reducing a little bit the image quality. So to be, let's say, in the first scan, a little bit faster. So I was introduced in the beginning. I'm an advanced workflow specialist for widefield imaging and not for confocal imaging. So I also have no clue about setting up confocal images. And I don't need to have that. If I now click on live in this very same position, we switch to confocal mode, and Mica is again setting up all the 4 individual laser diodes and also all the 4 individual hybrid detectors that are operating and recording here the image. Okay, so Mica is again setting up the image quality. And we are going here to maximum. I don't want to have the maximum image quality, as I said. I want to have live. Thank you. All right. This is what I wanted to do. So at the very same position you see here around, this is still the view that we have with the widefield system. And now we are doing really a true point scanning confocal image. So really scanning pixel by pixel in that image with a full confocal beam path. All right. So you can switch easily by simply clicking. If I now go back to widefield, all right, I can go here to live, and I'm back in widefield mode. All right. And if I now click on confocal, I click on live, I'm back in confocal mode. Okay. And not even this, as I have shown you already briefly, Also, the user interface stays the same. It doesn't matter whether you are doing widefield imaging or confocal imaging. No setting of detector windows, no setting of laser intensities to get everything out. What you do is you specify what kind of image quality you want to have, and that's it. What you can also do is you set up the field of view. For example, here I can specify now really the frame down to the position here of that cell, so to make it smaller, and increase, for example, to confocal grade or lightning grade, which is the highest resolution that we can achieve here with our system. And then I want to maximize the image quality. So now Mica again automatically adjusts here the imaging parameters. All right. And then what I'm doing once it's done, all right, I'm going to the position. No, this is the top. All right, where it was super tricky over here, maybe. Okay, like this. And then I tell the system, okay, looking at the time, we also want to do something else. So I go here to 3 micrometers. So now I tell the system around that current focus position, please do a volume imaging of 3 micrometers. Okay, so now we are really wanting to go into the exact details of the cell division event over here. All right, once this is done, I improve a little bit the imaging quality, and then I click on start. Okay, and this gives us, if we have a close look down here, we are recording now 20 Z planes, 4 colors again simultaneously imaged, and it's estimating the remaining time. We will be done at 10:41, so 4 minutes. This gives us the time to answer some questions in the chat, if there are any. And if you have forgotten, so you can also paste your questions in the chat if I have been not clear on some kind of things that I tried to explain. Do we have any questions that we can briefly address? If you're talking to me, I can't hear you.
Hi, Falco. It's Christoph here. I have to jump in because Jan does have some problems with her internet connection.
Okay,
but I just checking the chat functionality, and I see that there are no chats, no questions at the moment.
Okay. This, Jen, as a reminder to the audience, please go ahead. If there are questions, simply ask them, right? I mean, this session is also for you. I have seen the system already. So thank you for letting me know. So if there should be any questions popping up, just yeah, interrupt me or give me a note that I can address them also, that we spend some time on this. All right. In the background, no, wrong arm, this one. In the background, the recording is still running. And while this is finishing, I can also show you other results that I've recorded beforehand, because in the beginning, due to the stage problems that I had, I thought, okay, maybe I load a few other examples. So what we are going to see now is a confocal Z stack that basically looks like this. So here we have recorded a Z stack. It's actually a quite similar situation, right? Where in the beginning, this was the raw data set that we recorded with widefield, all right? And then we recorded here, that's that one exactly. No, that's the other cell, sorry. Where is it? Yeah, that's that one. Here we recorded the same situation. This is the thunder image, okay? So still widefield. And then we had a look at this very same structure, left-hand side also in confocal. And this then here is also confocal super resolution. So this is something that you can also achieve with Mica. We are not stopping at thunder imaging or confocal imaging. With this increased resolution recording that we do of the Z stack that we are also currently imaging, we can afterwards, with specially developed deconvolution, also increase the resolution further from the confocal image. And if you can have here a closer look, hopefully you can also appreciate. So the confocal image is already, compared to the thunder images, nice, crisp, and sharp, but we can go even further, okay? That you can really see substructures. You even see that Höchst also labels the DNA inside of mitochondria. It's really amazing. And then if you go down, And this is where confocal imaging is still the go-to method. When you want really to resolve the individual fibers here of tubulin, you need to go to confocal imaging. Okay, and as I said, this right now is simply a cell culture sample or specimen, but you could also achieve this in a tissue section or organoids, for example. And talking about organoids, this is something that I usually want to do is I show you some application examples that I've recorded with other customer samples, just to give you an idea of, let's say, more three-dimensional specimen. All right. So what I have here, or what I brought here, is, for example, an organoid. An organoid, and this is already the confocal image, but I want to show here first of all organoids that are labeled with M cherry, so H2B M cherry, so a nuclei marker in red and in green. Okay, and this was recorded in a 96 well plate. And as you can see, or hopefully appreciate, this is a wide field image. All right, and where you would say, okay, in order to see now really structure in the organoid, the wide field image is not good enough. But on the right hand side, I'm now loading the thunder image. Okay, and as I said, thunder removes the out of focus blur from your sample. Okay, and there you start seeing that we can also have a look from the bottom of the organoid to the top. Okay, that's, I think, already quite nice. And in theory, we should also have an example where I have the confocal pieces at the very same position. Let's see, that's not the case. And the last sequence here, yeah. So that's here the thunder image, and then on the right hand side I can load the confocal image stack. So that's the cool thing, right? Just imagine you are scanning a 96 well plate. You have 96 wells where there are organoids inside. So you are very fast in screening those 96 wells and using the wide field or thunder mode of Mica, because as I said, you can image up to 4 channels at once. Okay, something classic like blue, green, red, and far red. And then if you have specified, in well, I don't know, D7, there is a pair of organoids that I would like to investigate further. So you go there, you do a thunder stack, and then you see, the image quality is not good enough. I would like to have more resolution, and then you switch to confocal. So you're not doing all your imaging with a confocal microscope. Compared to any wide field system, confocals are simply, due to their way of imaging, doing point by point for the image pixel. They're simply slower, okay? And you should only use a confocal microscope when it's really necessary. A, because of the resolution that you need, or B, because of the three-dimensional representation that you need in order for your analysis. Okay, and in this case, you would say, yeah, the image quality was simply not sufficient, and you can also see how much more you can resolve with a confocal, those kinds of nuclei. Okay, good. So a three-dimensional sample. I also brought something else. And what I also brought is the other way around. I told you now everything about fluorescence imaging. But what about histological imaging? Like, for example, if I open up this image over here, what you can see here is an individual position of an H&E stained mouse embryo from a cross section. Okay, on the top left-hand side, what you can see is the overview that I have created with Mica in the beginning, right? So I have a slide where I have 5 individual mouse embryo cross sections, and I do an overview to select, okay, which section is the one that I need, because I need to have a certain position in an organ, whatever. And then you have this overview. All right. In this particular region of interest, or in the section of interest, you then record an image. For example, this one. Okay, looks like muscle. I don't know. And maybe this position, also something. Okay, you see some blood cells over here. All right, that's cool. And here as well, super nice resolution. But now somebody comes in and says, this is super nice. where did you actually record this? I said, yeah, here in this section. And yeah, but where there? So can you show me the position? Because exactly this kind of, I don't know, state I would like to see and have a look in the vicinity. How do you do this? Well, with Mica, what we now also offer is something that is called the context navigator. Okay, so what you can do is you mark your overview and the images that you are interested in. Then you have here a small icon that you can pop out. And what you see is the so-called context navigator. I make this now a little bit bigger. All right, so this is the overview. And of each of these sections, I have created high resolution images and then recorded those two images that I have shown to you before. And now what you can do is go to the zoom to function. And there are our two images, image 1 and image 2. And if you now hover over it, we are flying into the position where this image was recorded, or the other way around, where the other image, image number 2, was recorded. Okay, so you always can go back to the context of your sample. Okay, so it's a very convenient feature, especially when you're creating these gigantic overviews of, maybe you have slides where you have 50 sections on it, and afterwards you need to find exactly that position back because you need to re-image, you want to have a high resolution, whatever, and you are able to go back to this exact very same position. Yeah, that's something that I wanted to stress here very much. And in the meantime, of course, we are 10 times we're done here with our Z-stack. I don't want to hide this from you. So we are now going back. This is the Z-stack that we have recorded. This is the confocal stack. Okay, so confocal imaging with a high resolution. All right, nice confocal image. And now, as I said, and as I promised, what we can also do is lightning processing. You get the drift. So for widefield imaging, we have thunder as a functionality, and for confocal, we have lightning. And again, it should be as simple as possible. I simply click the apply lightning button. All right, and then we can have a look here side by side. On the left-hand side, we have the raw data set, and on the right-hand side, we have the data set with lightning, so with the increased resolution. All right, and you start seeing all the fine details in your specimen, even more than just the normal confocal image. All right, good. Asking to the hosts, are there now questions popping up in the chat?
So I have only seen one question, and this is a more technical one. So,
okay.
And I do like this question because the question is, how is it possible to do simultaneously images of 4 colors?
Yeah. Okay. Yeah. So I not immediately told you everything about it, simply because if there are also people in the audience that might not have so much imaging experience, I actually wait for this kind of question. So let me briefly explain to you how we achieve this. I have here my screen over here, and I also loaded up a PDF. And so let me answer your question by starting first with the classic situation. So we have our slide, so we have our sample, and in this sample we have 4 colors, all right? And these 4 colors, as you can see here in this representation, of course, their emission spectra are overlapping with each other, right? This is the classic situation, okay? And the problem of these overlapping emission spectra is simply that if you are imaging them at the very same time, you have crosstalk, right? So you see signal from one channel bleeding into the other one, and simply then you don't know, okay, which actually is the signal that I'm looking for specifically. So I'm looking for only the green signal, but how do I do this now? So what you do in a classic or conventional way is that you are using fluorescent filter cubes. So what you do is you look for an emission profile or a position in the emission profile where there's minimum crosstalk or no crosstalk at all, right? And then you specify this filter window to exactly that position, all right? So this might work here, for example, for DAPI, because you can go here a little bit more to the left-hand side and you could actually be free of any crosstalk. But what about the green dye, right? The green dye, if you would excite this simultaneously, would have the problem of massive crosstalk with DAPI, and also a little bit of crosstalk if you look at the entire emission spectrum with the RFP emission spectrum. All right. So what you do is you get more and more precise with your emission filter, but with this basically sacrificing fluorescence intensity that you capture, right, to be more specific. Because the most important thing is that you can be 100% sure that the stuff that you record is really only green, okay? And this is what we do in everyday life, right? So we are sacrificing fluorescence intensity. Now, if you consider this for 4 individual fluorophores being imaged at the very same time, you need to be very specific in your detection windows, and this makes it basically impossible to really get rid of all the crosstalk. With Mica, we are now doing something that, or we are introducing a technology that is called FluoSync. FluoSync was developed by Leica and the lab of Scott Fraser in the United States, who first came up with that idea, and we then worked together to create it, or to create a usable microscope system out of it. And what we are doing is, as I have explained in the beginning, we typed in the dyes, right? We typed in the dyes, and with the dyes, Mica knows what kind of emission profile are visible when it excites the sample. So you have here then the excitation of DAPI or Hurst in our case, GFP or 488, and so on. All right. And you have the very same situation. You excite all fluorophores and you have maximum crosstalk of all the emission spectrum. And this is something that we are not afraid of. Actually, the other way around, we embrace crosstalk, because by knowing the exact emission spectra of those dyes that you have specified, we can unmix those 4 dyes from each other, okay? And it does not only have the advantage that you can basically excite all the fluorophores at once, but with this technique, we are also able to record the entire emission spectrum, because we can separate all the blue photons or the photons that were emitted from DAPI or Hurst from the GFP or 488 photons, right? And the cool thing is that with this, our detection of light, or the amount of light that we are detecting, is getting a lot more sensitive, or the amount of light that we are capturing is much higher than you would only capture here a fraction, all right? And of course, we're getting fast. So basically, and this is also why I usually talk about it like it's a joke, but really it's the most critical point is that you are typing in the correct dyes, right? It's important whether it's DAPI or Hurst. It's also important whether it's RFP or Texas red, right? Because those emission spectra are not just red, but they are in their profile, they are distinct from each other. And this is what is important and what is needed for Mica to operate. And that's also why I said you could actually download further emission spectra to teach Mica new colors. Okay, as long as you can excite your fluorophores with UV light, green, red, or far red, you're perfectly fine. All right, so that's actually something that, so this live or so on the fly dye separation based on the emission profile is the cool thing that is basically enabling Mica to operate at that speed with that precision and to simultaneously record 4 individual fluorophore populations. And something that I just wanted to say was, it's now out of my head. In the meantime, if you want to read more about it, there is also a so-called white paper. So where we explained a little bit the technique, what is happening, because there is an intermediate, of course, calculation step that we use. But in order to, we explain a little bit how this technique is actually working. And in the end, you can also go to the original reference where this was explained in Nature Methods, how this technology is working. So, now I know what I wanted to say. Let's say the situation that I've right now explained to you is the perfect textbook situation. But if you are experienced with fluorescent imaging, and especially also with using DNA detection with DAPI, you know that over time, the emission profile of DAPI does shift to the red or to the longer wavelength. Okay, so the DAPI emission profile will look this when it comes freshly out of your fridge, right? But the moment you put it or you introduce it to your sample and then it's sitting there for a few days, this emission spectrum is actually moving more to the right. So actually the crosstalk with green is getting more. And there's an intelligence built into Mica that allows you to measure the emission profile of the fluorophore that you have specified in your sample. So in your very particular chemical or physiological environment, your dye might behave a little bit differently, all right? And Mica is able to detect this, and you as a user are able to adjust the detection of Mica to this specific specimen, so that you are always absolutely sure that the channels that you are recording are reflecting the reality in your sample. All right. Is there another question coming up? Ah, wrong camera.
Yeah, so this was very clear, and this was also a comment in the chat, so that it was a very clear and nice presentation. So thank you for that. Mm-hmm. It's also important to mention this. Yeah, we received further questions. So sometimes we need to look at materials, PVP, etc. Is the system able to image these well also in combination with the cells?
Mm-hmm. So basically what the problem, I assume, is that you have also a certain amount of autofluorescence that you are struggling with. Because if you have certain materials that are where your cells are growing on, it might be that they in itself, when they are excited with fluorescent light, that they can also emit light. So, and there is a certain way which might work also then with these specific materials, because we don't distinguish whether autofluorescence comes from a certain chemical or a certain material, or whether it's autofluorescence, for example, of chlorophyll, right? So if you are imaging a plant sample, you have a lot of autofluorescence. So basically you have your red dye or your green dye, and next to that is a chloroplast, and it's just shining bright. So what Mica can also offer is that instead of selecting the fluorophore, you could also select a fluorescent, dye, an autofluorescence channel. Okay, so Mica comes by default also with emission profiles for chlorophyll A, chlorophyll B, and as I said, if you are able to, with a different system, to record the emission profile of your material that you are working on, you can actually have this channel as one of the 4 channels that you are imaging. Okay, so this is maybe something that I should also make very clear. The detector unit of Mica, you know, I now demonstrated a 4-color imaging experiment. So we have a detector unit that consists of 4 individual camera sensors and also 4 individual confocal detectors. And this is, let's say, also right now the practical limit of how many different channels you can record simultaneously. So if you would select an autofluorescence channel, whatever that autofluorescence might be, you should be able to separate the autofluorescence population from the fluorescence population. But you are also here sacrificing 1 channel. Okay, so then your maximum amount of fluorophores that you introduce into the sample would be 3, all right? Or if you have a plant sample and you have both versions of chlorophyll A and B, then you only have 2 channels left. So that's simply a physical limitation, because the system right now only has 4 detectors, right? And then this means 4 detectors, you have 4 channels available. If you would have a system with 6 detectors, it would be able to separate 6 individual populations. But this maybe brings me back to the point I made in the very beginning. So Mica is not intended to be the one system that does it all, okay? So there are so many different applications that are going much, much more into the advanced field of imaging workflows that where Mica is simply not intended to. So what Mica can provide is a very easy and flat learning curve, okay, to do multicolor experiments in multi-well dishes or in multi-well plates and dishes and glass slides with any kind of biological sample or also grown on different meshes or whatever. But special applications like stat imaging, fluorescence lifetime imaging, turf microscopy, those are advanced imaging techniques, or we consider them as advanced imaging techniques. And for this, we have our dedicated platforms like Thunder Imagers or Stellaris as a confocal platform. And this would always be my suggestion. If you are struggling a lot with autofluorescence, it can be from different tissues or from different materials. What is really nice is our Stellaris platform, because there we offer a pulsed white light laser. This is now getting very technical. I put it very briefly. With this pulsed white light laser, you are actually able to separate the photons by their fluorescence lifetime, meaning by how long did it take actually on average for these photons to being emitted from the sample. And usually materials or autofluorescence is very fast, and your fluorescence is in comparison very slow. And this is then being used to separate photons from each other. I promise you it's a more advanced technique, but also they're very easy to use. And if Mica should not be able to remove or to separate the autofluorescence, this is then also a way to go. What I want to say is it's always nice to test things, and that's also why we do the session here today. So if you want to have a look with your specific samples, get in contact with your Avantor person of contact, or contact us directly. We are all working together here, so to provide you with also your personal experience if needed. We can also do remote demos, as I did right now, simply with your own samples. You can send them to me and I do the demos. So that's what we also offer for you to have more insight into the technology. All right, looking at the time, it's 2 minutes past 11. Are there more questions that we need to answer? If you have posted a question of what I wanted to say and you need to leave now, thank you very much for attending today. If you have more time, I can also answer more questions. And if there are any questions left unanswered, we will directly contact you via email and then answer those for you. Good. Are there any more questions?
No, there is only one question left. So is it possible to connect the Mica to a printer? So quite simple. The answer should be yes, you can do it directly on a computer, because there is no need for a direct connection. Or
No. So a direct connection of the system. Anyway, all the data that is coming from Mica is going directly into the workstation. Okay, that is also part of the package. And of course, there if you can have a Wi-Fi connection, Ethernet connection to a printer, USB connection, whatever. Yeah. So that's easily possible.
Okay. So further questions will be answered via mail after the session. So again, a very big thank you to you, Falco, for sharing your expertise and insights with us today on this innovative new system, the Mica. Yeah, it has been an incredible informative session, and we hope you all found it valuable too. So that's we would also like to remind you that a recording of this webinar will be sent to you shortly, along with a certificate of attendance. And you can also access this and other webinars on our website under the event session. Finally, we would like to thank all of our attendees for joining us today and for your engaging questions. We value your feedback and encourage you to fill out the survey at the end of the session. We look forward to seeing you all on our next webinar. So again, thank you very much, Falco and Leica, and wish you all a good rest of the day. Thanks a lot.
You too. Thanks a lot. Bye-bye.